Hey guys, my name is Shreyas and welcome to Simple Biology. In this video, we're going to be we're going to start introducing membranes by talking about the structure of a membrane. So, the first thing that you have to know about membranes is that they're actually made up of these small subunits called phospholipids. Now, if you've never heard if you've never heard of what a phospholipid is before, I'd highly recommend that you go check out our lipids video where we talk about phospholipids in a very extensive manner. And I think that just watching that, we talk about the chemical structure of phospholipids and just all the lipids. And watching that video might help you have a better understanding of membranes. You know, in biology, what ends up happening is that we start to see like topics which we learned before start to build up. You know, we learn small concepts in chemistry, and they build up to concepts in biomolecules, and those concepts lead up to cells. So, you know, don't have any gaps in your knowledge. If you don't know anything about phospholipids, I definitely recommend you go watch our video on lipids in our biomolecules unit. Okay, so anyways, just as a review, we know that I have a phospholipid molecule on a molecular level right here. We know that a phospholipid consists of two fatty acids. The two fatty acids are right here. Actually, let me use a different color because blue on blue looks kind of weird. So two fatty acids are right here. Okay. And then we have a glycerol. So the glycerol is right here. And then finally, we have the phosphate, and the phosphate group is right here. Now, from our biomolecules unit, and just, you know, whatever chemistry knowledge you know, you should know this. If you don't, then again, go check out our complete biomolecules unit. We talk extensively about this, and especially check out this video. But anyways, we know that the fatty acid chains are actually nonpolar. They are nonpolar because they consist of just carbon and hydrogen, you know? Sometimes you also refer to them as these fatty acid chains over here as hydrocarbon chains or hydrocarbon tails or chains. And we know that they're nonpolar. So why is that important? Because they are nonpolar, they do not mix well with water. They do not mix well with water. Okay? Now, in comparison to that, in contrast to that, the glycerol over here has oxygen atoms over here. So that makes the glycerol polar, and it does mix well with water. It does mix well with water. The same thing with the phosphate. The phosphate group, since it has a charge, it has that 2- charge over here, it also mixes well with water. Okay, so what that means is that it ends up, the phospholipid actually ends up having this difference in whether or not it mixes well with water. And that term is called amphipathic. Okay, so an amphipathic molecule is a molecule which is both hydrophobic and hydrophilic. Okay, hydrophobic is just a fancy way of saying does not mix with water. And hydrophilic is just a fancy way of saying does mix well with water. Okay, so an amphipathic molecule is a molecule which is both hydrophobic and has a hydrophobic region and a hydrophilic region. So in terms of a phospholipid, we notice this here. We have the hydrophobic region right here with the fatty acids, and then we have the hydrophilic region right here with the glycerol and the phosphate groups. Okay, so why does this matter to anything with membranes? So membranes actually form when the phospholipids come together to create something called a lipid bilayer. Let me write that down for you. Lipid bilayer. So if you look at this diagram over here, we can see that this is there are a bunch of phospholipids kind of just grouped together in like just all massed together, right? So this is actually a lipid bilayer. This is a membrane. We have water on this side, on the outside, and then we have water on the inside. So what's going to end up happening is the phospholipids are going to kind of arrange themselves in a manner like this. So if you look at our molecular diagram, this part this part right here is the head of the phospholipid, and that's everything that you're seeing right here. And then this part right here is the tail of the phospholipid, and that's what everything you're seeing right here. So since the head of the phospholipid is hydrophilic, it's going to be on the outside, right? Because it mixes well with the water over here. And then since the tail of the phospholipid is hydrophobic, then it's going to be on the inside over here, right? Because it does not want to mix, it does not want to touch the water. That's why we get this kind of this lipid bilayer structure like this. Okay, so I hope that makes sense. Now let's go and talk about phospholipid movement. So phospholipid movement. Phospholipids have lateral movement. So lateral movement is side to side. You know, as you can see here, it's kind of just switching back to forth on the same side. And this happens very frequently. So the lipid bilayer as a whole is not actually stagnant. It's constantly in motion because all the phospholipids are rapidly switching places back and forth like this. Okay, this is called lateral movement and it happens all the time. However, in contrast to that, phospholipids rarely switch sides. They never go, you know, this phospholipid and this phospholipid are never ever going to switch sides. This does not happen very often, okay? As you can see here, that's called a flip-flop motion, and as you can see, I wrote that it happens only once per month, so it's very, very rare. However, if a phospholipid actually does want to flip sides like this and go through the flip-flop motion, the flipase enzyme can enable it to do that. Okay. Now, in terms of fluidity, so what is fluidity? You know, whenever we talk about liquids, we can talk about how fluid or how viscous they are. For example, we know that water is a lot more fluid than something like oil, right? So that's an example of fluidity. So when we talk about lipid bilayers, we also talk about fluidity. It's very, very important. Okay, so fluidity can be the fluidity of a lipid bilayer depends on many things. One, it depends on the type of hydrocarbon tails that the phospholipids have. Okay, so if you have unsaturated hydrocarbon tails, remember that we talk about this too in our lipids videos, so you might want to go check that out. But an unsaturated hydrocarbon tail is one which has a double bond in the whole chain, and that's going to create those kinks, right? So you don't have a straight hydrocarbon tail like this anymore. You're going to start to see these kinks, the hydrocarbon tails kind of just jetting off like this. So what does that mean for the lipid bilayer as a whole? So what's going to end up happening is that since the lipid bilayer is the lipids within the phospholipids within the lipid bilayer are not able to come closer together because of all these kinks, it's going to be more fluid, okay? Because you can kind of imagine it's not compact. They're kind of all like drifting apart like this because of these kinks. So they're all the lipid bilayer as a whole is kind of like fluid. It's like a very, very smooths very well. It flows very well, okay? And that happens again when we have unsaturated carbon tails within our phospholipid. Very important that you know this. In contrast to that, whenever we have saturated hydrocarbon tails, this is hydrocarbon tails which have no double bonds between their carbon, so they're just kind of straight like this. As you can observe here, they're all just straight going up and down. That's when that allows for the phospholipids to kind of like all bunch up like this. As you can see here we have all these spaces between the phospholipids, but here there's no space. Everything's kind of bunched up together. It's very compact. So since it's so compact, what this does for the lipid bilayer as a whole is that it makes the lipid bilayer very viscous because all the molecules are just kind of compact, kind of pressed together. Okay, so that's one factor that affects fluidity. Another factor that affects fluidity is temperature. Okay, just to give you something you can relate to, you know, you never ever want to leave chocolate. You never ever want to leave chocolate in your car, right? I don't know if you've ever done this, but what will end up happening is if you do that and you leave it out on a hot day, as the temperature of your car begins to rise, the chocolate will melt and it'll end up leaving that big, like a big stain, right? You know, the chocolate will go from solid to maybe like a kind of like semi-solid to a liquid, right? So in the same sense, a lipid bilayer also has this property. Okay, so whenever the temperature of a lipid bilayer increases, its fluidity increases, and whenever the temperature of a lipid bilayer decreases, then the fluidity also does decrease. And this is called a direct relationship. You know, that's kind of from math. Okay, now So that's fine. The thing is that sometimes cells run into this problem with temperature and the fluidity. Okay, so in the summer, in the summer, we know that in the summer we have high temperature, right? So what does that mean for the lipid bilayer or the membrane itself, right? So whenever you have high temperature, you have a very high fluidity. The fluidity is high fluidity, which is bad for the cell itself, because what's going to end up happening is you have your cell, and then you have the cell membrane, right? And you have your nucleus. I'm just going to draw that in. But what will end up happening is, because the cell membrane has this happens during the summer when you have the high temperature, since the plasma membrane has end up becoming so fluid, it's not going to be able to contain everything in the cell anymore, because it turns into a liquid, and then everything just kind of falls out, which is bad for the cell, because if everything falls out, then the cell can't function, right? So that happens during the summer. Now, in contrast to that, in the winter, the opposite happens. Okay, so you have a low temperature, and then you have viscosity, or low fluidity. Same thing. Low fluidity. So what does this mean for the cell membrane? So what it's going to end up happening is that the cell membrane is not going to be it's not going to be a liquid like this, but it's going to actually become very, very, very compact, very, very compact. And it's going to turn into kind of like a solid, so which is bad for the cell, because if the lipid bilayer becomes a solid, then nothing can pass into the cell and nothing can pass out. Okay, if the cell is rigid, it's very, very bad, and because of that, the cell will die. So this is an issue that all cells run into during the summer and the winter. And a solution to that, biology actually has a solution to that, and that solution is cholesterol. Okay, now cholesterol is only found in animals, and it's classified as a steroid. If you don't know what a steroid is, we talk about what that is in our lipids video as well, so go check that out. As you can see here, this is cholesterol with its 4 fused carbon rings, indicating that it is a steroid. So cholesterol actually opposes this temperature chain in lipid bilayers, and prevents this high fluidity or viscosity and low fluidity from occurring. So how does it do that? So let's first look at the case with summer. Okay, so in the summer, we know that the phospholipids are going to want to spread apart from each other. They're going to want to spread apart like this. They want to come apart like this. They want to come apart. Erase that. They want to come apart like this, right? And that's what's going to cause the lipid bilayer to actually become a fluid, because if everything is kind of falling apart, then all the molecules, all the phospholipids are not compressed together anymore. They're all far apart, and then your lipid bilayer ends up just turning into liquid and breaking apart, right? So that's going to happen in the summer. But what the cholesterol does, these cholesterol molecules over here, is they actually prevent the phospholipids from getting far apart, because they kind of hold everything together. You kind of think of it as a tug of war, right? Like if you imagine that you had like in a tug of war, you're getting pulled on either side, but the cholesterol kind of holds everything together. It kind of grabs all the phospholipids together, and it prevents the phospholipids from all spreading apart. Okay, and it's going to keep the phospholipids close together, and prevents the lipid bilayer as a whole from turning into a liquid in the summer. Now, opposite to that, in the case in winter, so in the winter the opposite thing happens. All the phospholipids want to compress together like this. Okay, they want to come closer together. Okay, they want to become compact. And that's bad, because again, it's the lipid bilayer is going to turn into a solid if that happens. So in order to prevent that, the cholesterol, as you can see, it's kind of in the way, right? So it's going to prevent all the phospholipids from compressing with each other by just kind of being in the way and pushing the phospholipids apart from each other, therefore maintaining that balance between the high fluidity and the low fluidity, the balance. Okay, so that's all you have to know about membrane structure for now. In the next video, we're going to be talking about proteins which are found in membranes. But that's it for now. The membrane is as simple as that.